Hello students, I am Nupur Shree, Assistant Professor, Department of Psychology, Patna Women's College, Autonomous. Today, we are going to talk about how the communication happens within the neurons. Neurons are electrically charged even when these are at rest. A neuron is at rest means that it is not currently sending a neural impulse or message to another neuron. And when it becomes active and initiate the process of sending a message, we refer it as cell firing. There is a semi-liquid jelly-like solution inside and outside of the cell in which there are charged particles or ions. The cell membrane itself is semi-permeable, which means that some molecules may freely pass through the membrane while others cannot. Some molecules may only pass through tiny protein openings or channels. In the diagram, you can see the round structures over the membrane. These are the ion channels. But many of these channels are gated, or in other words, they open or close based on the electrical potential of the membrane. Although both positive and negative ions are located inside and outside of the cell, the relative charge of ions inside the cell is mostly negative, and the relative charge of ions outside the cell is mostly positive. Now, which ions are present inside and which are outside? Before discussing the presence of ions inside and outside, we should know that there are two kinds of forces that act upon these ions: force of diffusion and electrostatic pressure. Force of diffusion pushes these ions from higher concentration to lower concentration, and electrostatic pressure repels the like-charged particles and attracts the opposite-charged molecules. Now, coming back to which ions are present inside and which are outside. Inside the cell has a concentration of both smaller sized positively charged potassium ions and larger negatively charged protein ions. Outside the cell are lots of positively charged sodium ions and negatively charged chloride ions. The negatively charged protein ions, however, are so big that even if force of diffusion tries to push them outside, they can't leave, which leaves the inside of the cell primarily negative when at rest. Force of diffusion tends to push potassium ions outside, but because the outside is more positively charged, the electrostatic pressure doesn't allow to pass through it. In the outside, both sodium ions and chloride ions tend to enter inside due to the force of diffusion, but they can't enter the cell membrane when the cell is at rest, because the ion channels that would allow them in are closed. But because the outside sodium ions are positive and the inside is more negatively charged, electrostatic pressure makes the sodium ions to cluster around the membrane, waiting to enter inside once the specialized ion channels open. This difference in charges between inside and outside creates an electrical potential. When the neuron receives a strong enough stimulus from another neuron, the neuron fires. The cell membrane opens the sodium specialized channels one after the other in a chain reaction, allowing the sodium ions to rush into the cell. That causes the inside of the cell to become mostly positive because many of the positive sodium ions are now inside the cell. This electrical charge reversal is known as the action potential because the electrical potential is now in action rather than at rest. The action potential or in other words, the electrical charge reversal will start at the part of the axon closest to the soma, the axon hillock, and proceed down the axon sequentially opening ion channels. When it gets to the end of the axon, the message will get transmitted to another cell and the neurons come back to its resting state. Several things happen to return the cell to its resting state. First, the sodium ion channels close immediately after the action potential has passed, allowing no more sodium ions to enter. Small positively charged potassium ions inside the neuron also move rapidly out of the cell after the action potential passes, helping to restore the inside of the cell to a negative charge, and then the potassium channel closes as well. Once the neuron has returned to its normal resting potential, the sodium channels reset and the neuron is now poised and ready to fire again. However, the potassium ions leaving the cell results in a brief period of hyperpolarization where the membrane charge goes beyond its original resting potential. It returned to its normal resting potential when particular membrane structures come into play, transporting any remaining sodium ions back outside the cell and potassium ions back. When an action potential reaches a point on the membrane, the change in the voltage activates ion channels in a particular sequence. When a neuron is at rest, the inside electrical potential is around -70 millivolt. When a neuron is stimulated, the first to become activated are the sodium channels. They open slightly at first and sodium ions pass through the gates into the neuron. This causes the depolarization and the gates open a little further. As the depolarizing current kicks in, the gates will open fully and sodium ions will flood into the neuron. This creates the rising phase of the action potential. After around 0.5 millisecond, the potassium channels open and these allow potassium ions to flood out of the neuron. About 1 millisecond after the sodium channels have opened, the gate slams shut. The voltage across the membrane at this point is around +40 millivolt. Right now, only the potassium gates remain open and so the voltage starts to return towards its resting value. However, the potassium gates do not shut until the voltage has gone beyond and results in brief hyperpolarization of -80 millivolt. And after a brief period of hyperpolarization, the cell returns to its resting potential. This whole cycle, starting from resting potential to depolarization to hyperpolarization and again coming back to resting potential, is referred as action potential. And this is how transmission of the neural impulse happens within the neuron. There is a whole different series of events that are involved altogether in the transmission of message from one neuron to another. But for now, I hope you have understood the concept of today's class. Thank you.